This is another modified type of transmission electron microscope, which is called scanning transmission electron microscope (STEM). STEM is formed by addition of system that raster the beam across the sample to form the image. Raster is this method that an image is formed from the graphics or from small dots or from small dots. So the beam form this type of raster will form the image. The combined with suitable detectors, there are suitable detectors are present in scanning TEM, which form the image. The STEM uses magnetic lenses to focus a beam of electrons. So the type of lenses which all microscopes, electron microscope, are magnetic lenses, which are used for focusing beam of electron. The image formed is not by secondary electron as in scanning electron microscope, but by primary electron coming through the specimen. In scanning electron microscope, the secondary electron will form the image, that is the electron which come out from the specimen will form the image, while in STEM, the primary electron will pass from the sample, and this primary electron will form the image. Why in case of scanning electron microscope, we will study it later. In scanning electron microscope, secondary electron will form the image. Limitation of transmission electron microscope. Many materials require extensive sample preparation because a very thin sample is required, and it is very difficult to prepare a thin sample of all material, difficult to produce a very thin sample, and relative time consuming process with low throughput of samples. A lot of time is required for sampling. The structure of sample may change during the preparation process. When we are making a thin film, the structure of the sample can change during the sample preparation process, making the material in thin film form. Some materials are stiff, they can change the structure. Small field view cannot give the conclusive result of the whole sample. Small field view means only a portion of the material we can see, and the whole sample cannot be visible. Only portion of the sample can be visible in the STEM. Scanning electron microscope. This is another type of electron microscope, scanning electron microscope. An electron microscope that produces image or by sample by scanning over it with a focused beam of electron. Means the electron will not pass from the sample as was in the transmission electron microscopy. Electron pass from the thin sample, but here in case of scanning electron microscope, the electron not pass from the sample. They are scattered. Secondary electrons are produced. They are scattered from the surface, or they are absorbed on the surface. So scanning is done with a focused beam of electron. The incident electron interact with the electron in the sample, producing various signals that can be detected. So when the electron interact with the electron of the sample, various signals can be detected. Certain information about the sample's surface topology and composition can be obtained by SEM. Electron beam. The types of signal produced by scanning electron microscope include secondary electrons may be produced, backscattered electron can be produced, X-rays and light rays, that is cathodoluminescence, that visible light photons are produced by luminescence if the material is luminescent. A standard scanning electron microscope uses secondary electron and backscattered electron for image formation. Image is formed from secondary electrons or backscattered electron. While in case of transmission electron microscopy, we have studied that the image is formed from the electrons which are transmitted from the sample, and those electrons were primary electrons. Salient features of scanning electron microscopy. Electrons are used to create images of the surface of the specimen. That is topology. We can study the topology. We can see the image of the surface only. Resolution of objects of nearly 1 nanometer, 1 nanometer size can be visible. Small particle size having 1 nanometer can be visible in its resolution power is up to 1 nanometer. Magnification up to 500,000, 250 times greater than light microscope. 500,000 x. Its resolution is magnification is very high. Secondary electrons, base scatter electrons are used for image formation and are utilized for imaging. Specimen can be observed in high vacuum. In every type of electron microscope, vacuum tube, there is vacuum tube present. So the specimen are observed in vacuum. Because when the air is present, the electron will interact with the air and the image will not be clear. And high vacuum and low vacuum can be used. In environmental scanning electron microscope, specimen can be observed in wet condition. In environmental, because in the atmosphere there is moisture, so sample can be observed in wet condition. Scanning electron microscope gives 3D views of the exteriors of the object like cells, microbes or surfaces of inorganic material. It gives 3D view of the surfaces. STMs. There is another type of microscopy, that is STM scanning. STM scanning tunneling microscope. It was prepared in 1980. George Banning and Heinrich Rohrer invented STM scanning tunneling microscope, also called scanning probe microscope. Object resolution is, we have seen in the previous that resolution was of 1 nanometer. Here the resolution is even smaller, 0.1 to 0.01 nanometer resolution. Thin wire probe is used in this type of microscope, and this wire probe is made up of platinum iridium alloy. It is used to trace the surface of the object. It moves. This wire moves on the surface of the object, and the surface of the object is traced, and the image is formed. Electron from the probe. This platinum iridium probe. The electron of this probe overlap with the electron from the surface, tunnel into one another cloud. For example, this is the probe platinum iridium, and this is the surface of the sample. So this probe moves on the surface of the sample. The electron flow in between the sample and probe due to potential difference. This electron flow between the sample and the probe is called electron tunneling. And due to electron tunneling, the electric current is produced. Electron from the probe overlap with the electron from the surface, tunnel into one another cloud, because the probe is so close to the sample that electron tunneling takes place. Electron cloud overlap with each other, electron tunneling takes place. Tunnel create a current as the probe move on the uneven surface of the specimen. Here the current is produced and the probe move on the uneven surface of the specimen. So in different parts of the material have different distance. For example, here the probe will be more close, here there will be more distance between the probe and specimen. So in different positions of the sample, different amount of current will be produced, and on the base of this current, image of the sample will be formed. Application of STM. STM can be used in ultra high vacuum, air, water, various other liquid. Why we have seen that in SEM and scanning electron microscopy and transmission electron microscopy, only the vacuum is used, where the air, water and various other liquids or gaseous environment can be used, because the electrons flow between a very small distance, that is between the probe and a sample, and this atmosphere will not affect the flow of electrons. And a temperature ranging from near 0 to few hundred degrees Celsius, we can use a variety of temperature during its application. First, movie mat using STM. That is, we can prepare videos of the surfaces of the material, and the first video of the surface of the material by using STM was mat of what? Individual fibrin molecule forming clot. How in the fibrin is present in the blood, how the blood clot is formed using fibrin. This video was made by using STM. Live specimen examination can be done with STM, as in virus infected cells exploding and releasing new viruses. These things we can see live in STM. Visualization of intracellular changes we can see because we can make videos with STM. Scanning tunneling microscope and atomic force microscope. We have previously studied the scanning tunneling microscope. Here is more about scanning tunneling microscope. This is scanning tunneling image of the periodic area of carbon atoms on HOPG, courtesy of diagram of AFM. Atomic force microscopy. We study atomic force microscopy later. Here is this image. This image of the carbon is formed by STM, scanning tunneling microscopy. In scanning tunneling microscopy, a short metal tip is brought sufficiently close to the surface of the solid sample that their electron wave function can overlap and electron tunnel between the two. This is again the function of STM. When a potential is applied to the solid surface, the electron flow between the tip and the solid give a tunneling current in the range of pico to nanoampere. The magnitude of the current is very sensitive to the size of the gap, changing by a factor of 10 when the distance changes by 100 picometer. As the surfaces are rough and when the probe is moving on the rough surface, the distance between the sample and the probe changes, and due to which the amount of current flow will also change, and due to this change in current, on the principle of this change in current, the image of the surface is formed. Another type of microscope is atomic force microscope, which is the latest. Atomic force microscope is based on the detection of very small, of the order of nanonewton forces between a sharp tip and atoms on a surface. This is sharp tip is present in this type of microscope, atomic force microscope, and this tip is scanned across the surface at sub-nanometer distance. This tip moves on the surface of the material and distance is kept in the range of sub-nanometer, and reflection due to attraction or repulsion by underlying atom is detected. In some places the sample attracted, at some places the sample may repel it. So attraction and repulsion is recorded, which is done by the atoms which are present in the sample or specimen, and this technique produces atomic scale maps of the surface. This is map of the surface in atomic force microscopy, and this is the tip of atomic force microscope.